third monthly mini conference that WAS has put on. For 2021, Moz will be doing these every month. And for next month, we've got Medhat Nasser talking about his latest research on varroa and controls for varroa. And we'll have Sarah Wood from Saskatchewan University talking about her research with European foul brood and some things she'd like you to do to participate in her project. Today or tonight's presentations and the cost of our licensing fee for the Zoom webinar license is being sponsored by Oliveira's Honeybees. And we've got our speakers up and ready to chat with you in a moment. On my screen, I've got Michelle on my left and Jenny's on my right. You'll see them in a moment. I assume we always know that right here at the time when we're starting, there are a lot of people logging in. So what I'm going to do is, if I pull this off successfully, I'm going to share a screen and I'm going to run a little, while people are getting set in, adjusting their audio and other type of things, I'm going to be sharing a screen and playing a little 3-minute video that Oliver's Honeybees made for us. And it bears watching. It's really nicely done. Assuming I can get this to the point I can see where it's at.
You're Mr. Technician.
I know. All right. I had this thing running full blast earlier. There we go. Hold on. Should be coming through now, right?
Yes. No sound though.
Except I think you forgot to click the button to share
hold on,
audio.
Hold on. You're right. Well, I haven't done a whole lot of sharing videos yet, so let me try this once more. I got to check the little box here that says turn everything on to optimize. Okay.
Whoops, now you're sharing the wrong screen, I think.
I think so too. All right.
That's all right. That's all right.
As long as I don't get flustered. Should be this one. Nope, that's the wrong one. Yeah. Okay, is it coming through now or am I getting
It's still the wrong one.
It's all about the bees here at Oliveira's Honeybees.
All right, I'm an abject failure here.
No, you're doing all right.
You got the sound, but not the video that time.
All right, maybe third time's a charm. What's this thing saying?
It's because Jerry has 4 monitors.
Yeah, but I had it queued up and I lost it, so.
Do you want to try and play it at the intermission?
Yeah, let's do that.
Okay. All
right, I'm going to turn it back over to Michelle. Michelle?
Hi, I'm here.
Hi, so let me give a quick introduction. Dr. Michelle Taylor from the New Zealand Institute for Plant and Food Research Limited, Hamilton, New Zealand. Well, Michelle has 22 years of experience conducting bee research related to pollination and pest, disease, pesticides, and environmental monitoring. Her primary interest is colony health to maximize production. And we have a special connection with Ms. Michelle because she did her master's work with us here. So it's my pleasure to introduce you to our former graduate student who now has a PhD, Dr. Taylor. So Dr. Taylor, take it away.
Thank you, Jerry, for that beautiful warm welcome. I am Michelle Taylor, and I do work as a scientist at Plant and Food Research, and I work with this amazing group of inspired bee and pollination researchers. It's great to connect with all of you Americans, especially some of the ones I've been chatting with already. It's a special place in my heart simply because I've been there and spent a couple of years with you guys. Montana beekeepers, as some of you may have even taught me to keep bees, it's especially great to connect with you. You'll be pleased to know that I have stayed the course and I still love to conduct beekeeping research with actual beekeepers. So that's probably my main part of beekeeping. Today, it's my privilege to tag team with Jenny Walk, actually, and we're going to delve into the world of honeybee gut microbiome, and I'll provide you with an overview of the honeybee gut, focusing particularly on bacteria, and then I'll discuss some of our research, seasonal differences, and as well as the effects of diet and antibiotics on the gut bacteria. How's that going? That's clicking on my end. Can you guys hear me?
Yeah, somebody's clicking.
Okay. And after I've done that, we'll look at the impact that the gut microbiome. So then Jenny will take over from me, part 2, and she's going to look at the microbiome and how environmental factors such as pesticides and exposure and disease impact the gut microbiome, and then conversely how they might be influenced by the microbiome. So the microbiome, we're going to probably start with that one, and I probably should put up my slides so you can kind of see. Let's share that screen with you. Right.
There you go.
There we go.
Better than I did.
There we go. So the microbiome pretty much describes or defines pretty much all of the communities of microbiota within an organism. So, for example, a microbiota might be the communities of bacteria or fungi or viruses that live, say, on my hand or in your mouth. And so I guess the question is, why on earth then would, we want to look at the honeybee gut? Well, it's pretty much for the same reasons that we study the human gut. If we can understand both what resides in the gut and their interactions, and then the effect that that environmental factors might have on these gut residents by what, either what the bees are ingesting or either ingesting by food or other means, then we get to understand why they're there and what they're doing, and whether we can do anything to ensure that the bees are not only surviving, but actually thriving. So worldwide, we experience loss of honeybees all over. So that's known, it's well publicized and can be broadly sort of pretty much popped into 3 types of loss. The first being the slow incremental loss over time, which you guys have seen in the USA between, the late 1940s and the mid 2000s, where you've got this, they talk about a 59% reduction. Then you've got the second point, which is the unexplained rapid colony loss, which again you saw in the mid 2000s. And the third is what we all see, which is this annual colony loss. And that can range from anywhere from sort of 5 to greater than 30% loss, depending on where you are in the world. So in New Zealand, we have about 10% loss, and that makes it about 95,000 colonies. As far as I'm concerned, that's way too high. So what I wanted to do was delve into the honeybee gut and see whether there was any keys that the gut might hold that would enable us, regardless of what's going on in terms of our bees and us managing our bees, whether there's anything in there that will actually help us to mitigate some of these losses. So actually worldwide, we've actually seen an increase in honeybee colonies. And so we've seen that since the 1960s, and it's slowly increased and increased. And what that indicates to us is that beekeepers are actually really adept at replacing these annual colony losses. And that's why they're not noticed quite so much, because we just get in there and replace them. We lose 10% of our colonies to maybe Varroa or American foul brood or Nosema, as we see on the screen. But we don't talk about those so much because they're just sort of low levels, but they are occurring. So is there some key that we could look at to sort of mitigate those losses? And it all pretty much comes back to what Jerry drummed into me when I was in the States with him. It's that beekeepers are not beekeepers, the bees themselves are actually pretty much flying vacuum cleaners. So as they're foraging, the bees are picking up more than just their food. They're picking up nectar, pollen, propolis, and water. But at the same time, the mouth is also the ingress for antibiotics and pathogens and bacteria and pesticides and pretty much everything that's in the environment. And these factors are constantly changing with season. And so the digestive tract should really be viewed as a fluid and a changing system. So as scientists and beekeepers, we need to keep that in mind when we're doing any of this research. That should be pretty much fundamental. As these properties kind of move through the gut, they interact with the gut residents, and then the outcomes can be anywhere on the spectrum from beneficial to detrimental. So that's just kind of setting the scene a little bit there. And to clarify that, we've got our healthy bees in the middle. On the left-hand side, we've got our bacterial residents there. So they're impacted by the diet or what they uptake, but then also it's the pathogens that are having a massive impact, that could be having a massive impact on them. So some of these interactions have been proven and they're in the literature, which is great, and some of these are still hypothetical. So we're starting right at the basics and give you this brief overview, and then Jenny will sort of delve in a little bit deeper, but there is so much more that we're not going to be able to share with you tonight, but we encourage you to have a look into the literature and really get a good understanding about that. So now that we understand the dynamics of the honeybee gut, let's head to New Zealand, come and have a look at some of my beekeeping, and we'll zoom down into the gut bacteria so that we can discuss the basics of bacterial residence. So here we are, New Zealand. We're going to zoom down into where I am at Ruakura. And as you can see here, I'm going to fly in over our colonies, some of our colonies, and our cute little pink bee suit. No, it does not mean that the bees don't see you. And yes, they will still see you wearing a pink bee suit. So just keep that in mind. So as we delve into the bee gut, just as this clip suggests, we're going to start with the big picture, provide you with a context, and become more focused by the end of the evening for you. So just to give you a bit of a history, pretty much the understanding of this area is still developing really, really quickly, but the surface was scratched in the lab, back in the 1960s, by culturing bacteria from the gut on growth plates. But what that meant is that only the bacteria that was able to grow in those conditions survived. So now we have these great new techniques that allow us to identify not just bacteria that's suitable to the conditions of the growth plates, but pretty much the full composition that's within the bacteria within the gut. And we're starting to be able to link the specific bacterias with functions such as metabolism of sugars, pectin breakdown, pectin's the outside of the pollen, disease defense. And so now that we're aware of the major types of bacteria, we can use these, the growth techniques and some of these older techniques to quickly identify their characteristics and to feed into how these actually may work. And then we can take that into the actual colonies and look at those and run trials on them. So we're in a really exciting, accelerated phase now to see where we're going. So you've got excellent scientists in America, and over the last 10 to 15 years, they've contributed to this area hugely, and they've really set up this whole area of the gut microbiome and honeybees really well. And so they should really be, hugely congratulated and supported, because their research has laid a really sound foundation and has become a springboard for a lot of us to conduct our own research. So it's been amazing. So hang on a minute. Yeah, if gory pictures make you slightly queasy, then it's probably time to look away, but we're just gonna have a look at a dissected gut. To be honest, most of you all have gone seen this when you're out beekeeping. You get stung, the bee, dramatically pulls herself away and up leaves her gut sticking out with her stinger stuck in you. So you'll probably have seen it before. But what we saw, is what we know right from when I started my PhD back in the 2014, so a long time ago. We didn't know a lot. And so we did know, though, that there was about 1 billion bacteria in the honeybee gut. So it's quite a lot. And that these billion bacteria are broken up into sort of pretty much 8 to 10 groups. And the way I call it a group is we actually call it a phylotype. And what that means is that it could be an actual identified species, or it could be a family of bacteria that we know about. And we've got these really cool names. The beekeeping industry's got some cool names. You'll recognize Lactobacillus from human gut, and there's Firm 4 and Firm 5 of those. But there's also Gilliamella apicola and Snodgrass alvei, and then obviously the two dogs one, Bifido. Just really, Bifidobacterium is really the actual name, but we just call it two dogs. And the bacteria can be either detrimental or beneficial, but predominantly it's looking like they're really beneficial for us. And we also knew that newly emerging workers acquire this bacteria from their hive mates and also from the fecal deposits within the colony. So that's kind of what we knew sort of a little time ago. To give you a little bit more of an understanding, assume that the head is on the left-hand side there in the picture. The crop is the next piece, and this is not really technically part of the honeybee gut, but it's the interface between the outside environment and the gut. And so it holds the nectar that's coming in and is then put into the cells. And this has limited bacteria, but the bacteria that's in here is often associated with the plants that they're bringing back in. There is some with their own bacteria in there, but it's just limited bacteria in terms of what's actually honeybee gut bacteria. Then we move on to the gut, and that's comprised of the proventriculus, the ventriculus, and the small intestine. And this area is not that stable because the small intestine is like constantly sloughing off. The epithelial cells in there are sloughing off so that they can conduct the digestion. And so what that means is that there's not, again, it's still pretty limited in terms of the amount of bacteria that's in there. And so in that area there is where some of the absorption's taking place, and also where nosema is kind of sitting in that area there. And so the ileum is the next little piece along, and again that's sort of limited, but it's much more stable in that area. And then the hindgut is the rectum and the anus. So there's loads of bacteria in there in terms of quantity. You've got lots of lactobacillus and lots of bifidobacterium in there, and also you've got all your feces sitting in there.
Michelle, I'm going to interrupt you for just a second. I forgot to tell everyone, use chat for comments. I can't hear it, or it's fine, or informational, or hi, Randy, or whatever you want. Use question and answer if you're viewing this for any questions that come up, and then Dr. Hill will ask those questions of Michelle after she finishes her presentation. So I forgot to tell your audience that they can ask you questions through the Q&A. Sorry to interrupt you.
No worries. So throughout the presentation, we want you to sort of, you are beekeepers, but you also need to start realizing that you are actually keeping bacteria. So you're bacteria keepers as well. Not just within this presentation, but beyond this presentation. So if you're aware that the bacteria that's in the gut, what you're doing to your colonies, where you're placing them, what you're putting into them may actually be doing more than you actually realize, because the bacteria in the bees are responding to that. So that's a good thing to keep in mind for the whole of this evening. So when I started, so what I'm going to do here is when I started the PhD, I'm going to choose now 4 knowledge gaps that were there that I identified at the start of the PhD. The first was that there was no New Zealand data. Nobody had done any research on gut bacteria and honeybees. The second was that there was no information about seasonal differences. The third was, is there any effective diet? And then the fourth is antibiotic treatment. So I'm going to run through those, and then Jenny will delve into things a little bit more intensely. And the reason why it's important, so the literature states that because that there's 8 to 10 phylotypes, and that all bees have these. But the reason why I was a little bit sort of weary about this is because we have a closed honeybee population in New Zealand. And although it was established from imported Italian bees from Europe and subsequent imports from the United States, the New Zealand population hasn't had any more bees since the 1960s. And also that we don't use antibiotics in New Zealand to control any honeybee diseases. So antibiotic use is illegal. And the reason this might be important actually is because we send up to 35,000 1 kilogram packages to Canada every spring, their spring. So our autumn bees are heading up to Canada every year. So what we have in our bees probably has a really large impact on, or may have a large impact on, what's going on in Canada. So in 2014, what I did is I conducted probably the largest assessment of gut bacteria, and I looked at 4, it was all conducted within a 2-week bracket. And so I collected gut bacteria from 7 regions, as you can see on the map there. So I worked with 21 beekeepers, and there were 3 apiaries per region, and then 6 colonies per apiary, and then there was 20 bees that I took from each of the hives. And because the samples were collected within 2 weeks, even though we have a temperate climate, it's quite long. And so what that means is the bees up north are actually a little bit more ahead of the bees that are down south. And so that was kind of nice to see whether the regional differences would have any impact. At the same time, I got beekeepers to provide me with bees that they deemed from colonies that they deemed to be sick, as well as the healthy ones. And then for each sample, I extracted the bees and then extracted the DNA from the bees, the bee gut, sorry. And then what we did is we have so each bacteria has a gene called a 16S rRNA gene, and that's specific to each bacterial type or species. And so what I'm going to do is, that's the one that I'm going to talk about, and probably Jenny's going to talk about that one too, just to keep it sort of simplify a little bit. And we'll just talk about that for the rest of the season. But pretty much we use that 16S rRNA sequencing to identify the different types of bacteria throughout our samples. And what I found is that we had 27 phylotypes in New Zealand, but 11 of them were actually really common. So they were in all of them. Those other, the ones after 11 were pretty much at much lower quantities, but they were still there, just much lower. And they included the 5 core bacteria that I talked about earlier, Snodgrassella, Gallimiella, those ones by Fido. So that was good. So that does corroborate the research from the US and Europe, and indicates that, you know, even though there's been a sort of a 70-year gap between when we have brought bees over here, that the bacteria are actually probably pretty stable. I found that there were no regional differences, which completely surprised me, because our native flora up north is completely different to down south. But anyways, there were no regional differences. However, the 8 bacteria were specific to sick bees. And that was a really exciting thing for me, because it suggested that some bacteria may actually be potential indicators of poor bee health. So we just keep that in mind, and just that, yeah, so that the sick bees did actually have 8 more specific bacteria. So then what I did is I got some data from Nancy Moran's lab. They had done a trial in Connecticut. So Eli Powell has written up that paper, really nice little paper. That's the one about bees acquiring their bacteria from their hive mates. And we analysed ours together. And you can see the blue dots on the left-hand side. Pretty much what it means is that any dot and circle that's close together means that they are similar to each other. So you can see the blue dots on the left-hand side are completely separate to all of the ones that are on the right-hand side there. So all of my communities. And so again, we saw that the core bacteria were common, but interestingly that there was 2 specific to USA and 9 was specific to New Zealand. So there was a slight difference there in terms of our, they're subdominant, but they're common. So maybe we call them subdominant core bacteria, I don't know. But anyways, we'll just say, for example, that there are 9 specific subdominant core bacteria, and they're different to what we see in the States. So that was exciting as well. So slightly different. So I looked to see if the bacterial profile of the gut within the colony changed with time of year, because that's one of the reasons why there may have been a difference between the US data and my data. Their data was collected in autumn and my data was collected in spring. So I looked to see whether there was, you know, any difference within our colonies over seasons. And so in 2017, we assessed bees from the same 5 colonies across the 4 seasons, so winter, spring, summer, and autumn. And we found that, okay, don't freak out about this graph. The way that you read it is that the bacteria are down on the left-hand side, so they're all listed down there. And then each of the columns are what you see in terms of winter, spring, summer, and autumn. So it's pretty much a heat map. Any of the brighter colours mean that there's lots and lots of that type of bacteria in the bees from that season. And so what you can see here is that the bacteria on the left, the bacterial types are highest in summer, and then that are least in spring. And so keeping in mind that this is when the colony restarts with numerous young bees, there's a couple of options why this might be. And so I've just hypothesized, there's a bunch of theories in there that I have that might be the reason why. But what's quite neat is that Kisner Rover's also done a paper, they put out a paper in 2020, and they found a similar thing, that gut microbiota structure differs between honeybees in winter and summer. So they just did the winter and summer. But for me, I can see here that there's a very clear difference between spring and summer and winter and autumn. So even more detail to what they've suggested. But what they did show is that the diet contributes to the differences, which is really exciting. And it's pretty much the same angle that I was taking with mine. So what might be causing these over the season? And as a beekeeper, of course, it's going, well, the first thing that we'll think about is food, right? Because that's what our bees are doing, or you know, foraging, or they're not, you know, it's winter, so they're bedded down. So we wanted to look to see what, you know, the differences between these bacterial loadings and why that may have occurred. And what we need to consider is that our management styles are changing over the years. You know, we don't do the same things with our bees than we did, you know, 60 years ago, 80 years ago. You know, we used to just put our bees in one paddock and that would be it. Now we're moving them around for pollination. Now we're moving them around to catch a nectar flow. Now we're moving around just because we've moved house ourselves and we want to move them. So we move our colonies all the time. So it's completely different type of beekeeping now to what we had, you know, decades ago. So I thought it'd be really interesting to look to see what the type of sugar was, you know, when we're feeding, supplementary feeding, and times where we need to build our bees up really quickly to get into pollination, say, or we've taken a bit too much honey off accidentally before we've gone into winter. So we need to top up our colonies with some sugar. I wanted to look to see whether that type of sugar would actually have any difference, have any effect on the gut bacteria. And so what I did was in 2007, I conducted just a case trial in the lab just to see how sugar type affected the bacterial profile in the gut. And I compared 3 different types of sugars. And the first was sucrose. And sucrose is a disaccharide. So it's made from the 2 monosaccharides, fructose and glucose. The next one that I wanted to look at was invert sugar, and that contains the monosaccharides, fructose and glucose. And then honey has both the monosaccharides and the disaccharides, but it has more of the monosaccharides. So what we did is we used 10-day-old bees with a full complement of bacteria, so they're pretty much painted bees, put them into the colony, taken them out 10 days later, so theoretically they should have a full complement. And we caged them and then we fed them 6 different treatments. And I compared those 3 that I talked about, but I used, the honey that I used was actually Manuka honey. And the reason for that was because we know that Manuka honey has antibacterial properties, sorry. And that's beyond the peroxide activity that you would see in most other honeys. And so we got 2 different sorts of this Manuka honey. And then I also looked to see whether the components of Manuka honey, so methyl glyoxal in sugar syrup, and the precursor of methyl glyoxal, DHA, in sugar syrup would actually have an impact on the bees. And so that made up the 6 treatments. We had 9 reps for each of those, and the caged bees in the 7th treatment were actually placed back into the colony. So we took them out of the colony, caged them, and then we placed them back into the colony for the trial duration. So they're our controls. And then we looked at the guts of 5 of these bees. We extracted those and we pulled those. And the reason we had to pull them was because when we started, that was wanted to ensure that we had enough actual DNA that was extracted. It was extracted. And then we sequenced that using the 16S rRNA sequencing that I explained earlier. And so this non-metric multi-dimensional scaling graph that I'm showing you here was just a bit of a look-see to see, would there be any differences, would there not? And what it sort of appeared to be is that the ones that are in sucrose, which are circled on the left there, were slightly drawn out from those ones that were on the right, which is the sucrose poor ones. So pretty much honey, invert sugar, and the other invert sugars with the components of the honey. And the sucrose-rich diets, they were displaced along that horizontal line. So you look at the differences along the horizontal line, and they were slightly displaced. So to get a handle on this, if the one of the bacteria tends towards the same direction as one of those groups, then it's more likely that that bacteria is influenced by that treatment. So just overall, just giving you an idea of what's going on. So in this case, the sucrose diets produced communities that converged with Rhizobiaceae, which is the bacteria on the left-hand side there, just over there. And the sucrose poor diets tended to increase in Gilleyamella apicola. So we then conducted some further research and analysis. And what we identified is that the sucrose-rich diets increased the relative abundance of 3 subdominant core bacteria, so Rhizobiaceae, Acetobacteriaceae, and the Lactobacillus cunkii. And they actually decreased the relative abundance of Freshella pereira, which interestingly in itself is a gut pathogen and causes scarring in the gut. I don't have time to go into that now, but that's a really interesting bacteria causing scarring. So as it's still early days in the field of science, we don't know whether the types of sucrose that the colony consumes itself has any effect on the colony health or the colony productivity, or if there's anything we can or should be doing about this. But it's definitely something to be keeping in mind, because it's a relatively, well, you know, in the scheme of beekeeping and management of bees for millennia, this is a relatively new practice. So what does it do? What's happening with our bees? So the next question then is, well, what else do we feed bees that may have an effect on the gut bacteria? You know, if you just think about your normal beekeeping activities, is there anything else that you guys are doing that you think that might be having an impact? And so for us, if we think about our American foul brood, we control that using fire. So pretty much we scorch earth and we burn anything that has American foul brood. But when I was in the States, we would feed terramycin for American foul brood. So feeding those antibiotics to control AFB. And so it's an antibiotic, so you would assume that it would have some impact on the gut residents. So these residents, remember, are the beneficial bacteria. So before I wrap up and pass over to Jenny, I'll just take a quick look at whether antibiotics affects the gut bacteria. And so as you can see here, the newly emerged, so what I use is newly emerged caged bees, and I feed them either sucrose or I fed them with or without terramycin for 8 days. And they were compared against the control bees that were actually taken out from their cells 24 hours prior to emerging and then fed sucrose. So I analysed the data slightly differently for this one. This is using qPCR. And so pretty much what that means is it's just a total count of bacteria. So is there more bacteria in one sample than there is in another? And so no surprises, those that were fed terramycin were had less bacteria, but interestingly is that there's a huge variation in the sampling. So that confused me a little bit, because we don't use terramycin here in New Zealand, but we've got this massive variation. So some bees perhaps are susceptible, and some bacteria in the gut are susceptible and some are not. And so that was interesting to me in itself. The bee controls on the left, so these are the ones that have been removed pre-emergence and fed terramycin, had the least variation in bacterial counts amongst reps, 10 to the 6. So possibly moving forward, that's actually a really good way a good thing to use as a control. You know, bees that have been removed from their cells prior to emergence. So maybe there's actually something going on with emergence, because these bees were also fed terramycin for the time once they had been removed, and they were fed terramycin for the 8 days as well. So that's an interesting fact. The qPCR down a little bit down the bottom there, that just shows that there was pretty much no contamination. So the fact that terramycin reduces bacteria in the honeybee gut is not surprising, but interesting that numerous countries still feed their bees antibiotics. And so we probably want to have we don't know, but we do want to have a good think about what this is actually meaning when we're feeding antibiotics. What's that doing to the gut residents within our honeybees? And as beekeepers, this really is worth contemplating. I don't want you to go out and jump and stop controlling AFB. That's not what I'm saying. But what I am saying is I want you to think about how you're controlling your AFB. So this nicely leads into to Jenny's presentation about understanding how environmental factors such as pesticide exposure and disease impact the gut microbiota, and conversely, how the microbiome may play a role in influencing susceptibility to these factors. So I'll pass over to Dale, and yeah, happy to take any questions. So thanks for your attention, and I look forward to your questions.
Yeah, Michelle, thank you, Michelle. Great talk. you would make Colin proud of seeing the PCA analysis and how far you've come with using statistics nowadays. And he's retiring this year.
Oh, really? Cool.
Great. Can you stop sharing your screen so that we can see you again?
Yes, I'm just trying to find it. Let's see if we can find it. There we go.
The green button.
Oh, I know. Yeah, I just can't actually find the oh. There we go.
There you go.
Buried underneath everything. Sorry about that.
Okay. Well, it's my pleasure to introduce Dr. Dale Hill to everyone. Dr. Hill is an animal nutritionist. Like myself, he's supposedly retired. Also, like myself, he's not doing too good a job at retiring. He's teaching the nutrition section in one of our online courses. He just finished last week. Well, actually, he's still been doing that this week. But I thought he was an appropriate person to be the moderator for the question and answer. So Dale, do you want to take over?
Yeah, I guess the first question for Michelle, I've got a couple of really interesting questions. And the first one is, does the bee microbiome change with the role of hive activity or hive maturity? That is, do the nurse bees have different bacteria than the foragers?
So what we know is that the early bees, like when they've newly emerged, they don't have, there we go, that's probably better. When the newly emerged bees, they don't have well, they have limited amount of bacteria, and then they collect that from their nurse bees and their surrounding hive mates. And so then what happens when we go into the foraging bees is I don't have a good handle on that actually, but I'm imagining that so we saw a few differences with some of the ages of our bees, but I haven't published any of that yet. So.
Okay, another real quick question is, are some bacteria symbiotic like E. coli? And then how do you identify the phyla? By a DNA technique? And have eDNA techniques been applied to the hive in general to gain insight about what else bacteria or other lives in the hive, not just in the gut?
Yeah, so there's a bit of an overlap between the bacteria that are in the hive and the bacteria that are in the gut, but interestingly, not a lot. So you would think that, yeah, so in the gut, it's actually quite specific. Within, I think the number that sort of jumps into my head is sort of around about 7 to 14% is an overlap between the bacteria that you see in the colony and the bacteria that you see in the gut. There's a really nice paper that was done on that, but quite a few years ago now, maybe back in 2014 or 15, about the overlap of those. But yeah, they are not the same. So that's interesting itself. So what's in the hive is different to what's in the bacteria in the bees.
And then the supplements that are sold for honeybees contain bacteria not normally found in the bee gut. And if they do they compete with the bacteria that's normal or native to the bee gut?
That's a really good question. I've only looked at carbohydrates, so I haven't looked at protein and I haven't looked at the supplements that are being fed there. It was definitely something that I had on my list of things I wanted to do, but just ran out of time to do it. So I can't give you a good answer on that one.
Yeah, well, let me rephrase the question. Do the bacteria that you're feeding compete with the bacteria that are already in the gut, the native to the gut? Because they're not necessarily the same species.
Yeah, right. So they would be completely, so as we were just talking about before, the overlap between what's in the hive or what's in the environment is different to what's actually in the gut. So when it comes in, there will be interactions that are occurring between each of those. And because there are so many of them, I can't give you a clear prescription yet. Nobody can yet about which bacteria impacts on which, but there definitely are symbiotic relationships going on within the gut. So Gillymella and Snodgrassella kind of cozy up, and there's a few others that do that too. So, but then the interaction of what is coming into the gut. So when I was feeding sucrose, I'm just feeding sucrose that's sterile. So I'm not actually I wasn't actually feeding bacteria into the gut. So I can't, unfortunately I can't answer. It's still very new. Yeah, there's still, and probably somebody has done that already in the last year or so, but we haven't published it yet.
And then I guess one more quick question is, do we find these gut bacteria in the Asian honeybees, the Apis cerana indica?
Yeah, and even in just bumblebees as well, just in apis. So you see, yeah, but they're just different strains of them. So you'll see Gillymella in some of these in bumblebees, but they're just a different strain of it. And they don't, interestingly, if you take them out of a bumblebee and put it into a honeybee, it won't grow. And vice versa, if you take the honeybee one out and put it into a bumblebee, it won't grow. So they're specific to the type of bee that it is.
And one more question is, does the spring sugar feeding change the gut bacteria, or does it change when they start bringing in nectar?
This is a really interesting point. So yeah, we saw the differences between spring, summer, autumn, and winter. In that trial, we did not feed sugar syrup to make sure that that wasn't going to have any impact on the colony. So from that, I can only assume that it's from what they're pulling in from the environment and the changes that are going on with those bees over the season is probably more to do with the population size and the length of time that the bees live, probably more associated with that rather than actually, rather than, because I didn't see a difference between all of the bees throughout the whole of New Zealand, I'm imagining that some of those beekeepers were feeding sugar syrup because of the nature of the environment that they're in. So at that time of year, some of them would need to be feeding sugar syrup. So we can't see, there wasn't enough detail within the metadata that we collected to identify whether there was an impact of that. So that little study, the carbohydrate study, is an indicator that something's happening there and the food that they're taking is having an impact on the gut bacteria, but we can't pin it down fully to that. But it corroborates with Kesneroba's paper.
Jerry, how are we doing for time for questions?
We're doing fine.
Okay. As beekeepers, we are aware of the beneficial properties of Proplus as a benefit to the bees. My question is, does the Proplus harbor bacteria that is transmitted to the gut?
That's a really good question. That's a really good question. We don't know. We know that it's antibacterial, it's antiviral, it's anti pretty much everything. It's got all sorts of great properties that, you know, would make humans healthy. So that's fantastic for Proplus. Because it's antibacterial, I assume that it wouldn't be carrying a lot of bacteria in it itself. There might be a bit on the outside of the Proplus, but I can't imagine that there would be much in there. But Marla Spivak is a bit of a whiz on that sort of stuff. She, you know, look up some of her research and she'll probably have that answered in there.
Okay. Next question is, bees seem to like dirty water. Any carryover gram-negative bacteria from water sources to the bee gut?
Well, you know, when I said that the American bees had 2 different types of bacteria and the New Zealand ones had 9 different types of bacteria. Well, the 2 that actually were in the bacteria actually were, a couple of them were contamination. So, and they were bacteria that you would normally find in the ocean or that sort of thing. So it's, there is bacteria in water itself. Just sitting water always has bacteria.
Next one is,
but whether it has an impact, I don't know. It could have just been dead bacteria that the sampling picked up. The problem with the qPCR, or the qPCR2 and the 16S is that it doesn't tell you whether it's alive or not. It just tells you that it's there or that there's a quantity of it. Yeah.
Okay. Any comments on use of pesticides or fungicides and that impact on the bee gut biome?
I'm going to leave that straight for Jenny because she's going to be talking about pesticides and the impacts of the environment. So we're tag teaming, so she'll do that piece of it.
So with that, I think Jerry will go have Jenny start.
Well, I've got one question that came up in chat. So for those people, chat and Q&A are confusing. And it also depends on whether you're a panelist or an attendee, which one you can have access to. So I have one more question that came over on chat. And that was, Michelle, could you elaborate on the level of resolution of 16S RNA sequencing? You are resolving at the level of the phenotype or the bacterial species or the strains? That sounds like a very specific question.
Yeah. So what we do is we take, okay, I'll try and answer it. So we take the the DNA from within the gut, and then we sequence it. And so what happens is that sequence, so pretty much every gene, the bacteria have their own individual little genes. And so that gets a number, and that number is for a code, sorry, that code is in checked against a big database, that the national database. And so that database there has already been characterized, the bacteria within that, for that particular, in that database that's been coded there, has already had all of its characterization done. So we know that actually is a specific type of bacteria. It grows this long, it's this size, it's this color. And so that code there then transfers back over to my data set. And then I get that piece of data set. And so some of them are coded to right down to the species and even a strain, but sometimes they're only coded up at a family level. So it's sort of the big overall level. So in terms of what I'm coding for, it really does depend on the type of bacteria that it is. There's just thousands and thousands of pieces of information that you get once you've sequenced all your data, and then it's trying to make sense of that story based on what you have done and based on the codes that are provided by the databases. Hope that makes sense. But we also do cut off at a particular level. We have specific cutoffs that we use.
Okay, I think we'll stop the question and answers here, except for one thought, and that is, I see we've got Jenny and Randy, I know, who are people who might have specific questions. And since they are live panelists, I'm going to give them a chance for a question. Jenny? Randy, either one of you? There you go, Jenny.
Not right now for me.
Okay, Jenny?
Yeah, I'm curious if you presented the qPCR results for the antibiotic exposure. Did you do sequencing and look at the microbial communities for that, or no?
No, but I thought it would be a nice lead into yours because I know that you have, so that's good.
Yeah, exactly. And some of the questions that people had asked you, I think I'll be able to share some results at least on some of those questions too.
Okay.
Really great. It's really great to hear the work that you're doing there.
Thanks, Jenny.
Jerry and
Yes,
Michelle,
Randy.
I can say one thing with your question about the antibiotics. We just did a field trial and treated colonies with either terramycin 3 treatments or tylosin 3 treatments, and then gave them a dose of either of 2 different antibiotics. And then I took samples of the bees immediately after the antibiotic treatments. Then took waited a few days for the antibiotic to wear off, gave them a dose of probiotics, waited 7 days, took samples again, and then waited another 2 weeks for a full brood cycle and took samples again. All those samples now are next-gen sequencing, and we should be able to answer your questions about recovery from probiotics and relationship to recovery from antibiotics and relationship to feeding probiotics.
Fantastic. Yeah.
Thank you, Randy. All right, folks, I'm going to give one more shot at trying to play Ray's video because his crew did a really great job putting it together. Yeah, you would do that to me, wouldn't you? Let's see. I got all options on. There's Ray. Hopefully this will do it. And then once this plays, we'll go to Jenny.